So let's begin from the very first concept of today's class, fluidity of cell membranes and FRAP. Both we are going to discuss. So first, we are going to speak about fluidity of cell membranes. So from here till now, I was very fast, right? So I used to say what we are going to do, what and all, everything. So it will be fast for most of the students. That's fine. I'm going to discuss this fluidity of cell membranes and FRAP in detail and in much slower way. Okay. So if you have any doubts, I told you already, you cannot ask in real time. You have to mail me, or in Google Classroom, which I am going to share tomorrow. You can just add your doubts. Okay. So speaking about fluidity of the cell membrane. So people, most of us, we know the membrane, the cell membrane which is there, it's bilayer, and it is fluid in nature, right? Fluid mosaic model, we used to say. So this is fluid mosaic model diagram what I have displayed here in this slide. Now we know that it is fluid in nature, but we have to understand why it is fluid in nature and what are the factors which are affecting the fluidity of the membrane, and what happens if fluidity is lost, and how much fluidity the membrane is containing. That means it is too much fluid, or it is viscous in nature, or it is gel like crystalline nature, or it is very hard. It's not too fluid, it's not too hard, it's like viscous in nature. That means how much ever the interactions are possible, that much loose interactions or non-covalent interactions will be there in case of membranes, right? So that makes the membrane fluid in nature. So this fluidity, the viscous nature of this particular membrane, it allows major functions of the cell. For example, speaking about the functions of fluidity, right? So if it is because of fluidity only the cell can divide. The cell is going to divide into two in cell cycle, right? In mitosis, it is going to divide into two smaller cells because of the fluid in nature of the membrane. Next, it is important for sperm and egg interaction. So sperm and egg plasma membranes has to fuse, right? The fusion of any cell molecules, it is because of the presence of fluidity. That means just try to understand, if fluidity is lost, these are the factors, these are the things or functions will be lost by the cell. The integrity of the cell will be lost if fluidity is going to be lost. Try to understand this. That's why it's very important concept, right? Next. So speaking about other functions of fluidity, so cell signaling. So anything external stimulus, the cell has to sense it and it has to send the signal inside the cell, right? So complete cell signaling is based on the membrane fluidity. If it is rigid in nature, then signals cannot be transmitted into the cell. Although the ligand binds or hormone binds to its receptor, it is not going to be transmitted into the cell. Try to understand this. Next, so speaking about the different functions, endocytosis and exocytosis. These are exocytosis, endocytosis processes are not going to occur. Hence, it is very important. These are the few functions of the membrane fluid in nature, right? If it becomes rigid in nature, these are the factors the cell is going to lack with, right? So keeping this in mind, we are going to speak about what are the factors which are going to affect the fluidity, right? So this is the membrane. Try to see this. You have the structure, membrane, cell membrane structure. So this will be called as outer bilayer, OL, and this will be called as inner bilayer. That means inside the cell membrane it is there. So when I put a complete cell here, just go through this diagram. The outer layer OL and inner layer is IL. So this is how it is flattened here, the same thing. So outer layer which is interacting with the extracellular space, inner layer interacting with the cytosol. Next, so when you look into this structure carefully, so you have you have certain proteins called as peripheral membrane proteins. I'll just mark it, one. And second, we have integral membrane proteins. See here, we have integral membrane proteins, we have peripheral membrane proteins, and we have one more type of protein which is not shown here. I think it's called as GPI linked protein. The third one is called as GPI linked protein. Just remember this. Okay, so we have different types of proteins in the membrane. Remember this. Apart from protein, the major constituent of this membrane is phospholipid, right? So we have numerous millions of phospholipids that tend to cluster together to form a complete membrane, right? So these phospholipids are the major candidates which are playing an important role in maintaining the fluidity of the cell membrane, right? So we are going to discuss about membrane lipids, what are membrane lipids, how they are going to affect the fluidity in today's class. Next, speaking about glycoproteins, meaning the carbohydrate moieties which are attached to proteins called as glycoproteins. We have glycolipids, that means carbohydrate moieties which are attached directly to the lipid molecule, phospholipids. So based on glycolipid only, the ABO blood group will be blood group will be determined. So it's today's assignment for you. So just go and search about, after the class you just search about glycolipids and their role in A, B and O blood group persons. So how it is going to determine the blood group of a person, right? So based on this, in 2019 they have asked, December 2019 they have asked one question for 4 marks, right? Based on this glycolipids and their role in blood group determination. Keep this in mind. This is assignment, so you have to be serious in this crash course, then only it will be useful. And you have to take a lot of opportunity, this opportunity you have to take, and you have to consider this as your regular classes, and you have to follow the instructions which were given by me, right? So just go after this class, search, it's like assignment for you, what is A blood group, B blood group and O blood group person, and how it is determined along with the glycolipids. This is what you need to search. Okay, now, so anyhow, I'll be including this in Google classes. Okay, next. So one major factor what you are observing here is you have different colorations on the outer layer. See, just go through this. I'll just clear the annotations. Okay, so when you look into the outer layer, what is happening there? You have different types of phospholipid head groups. So you have red color, blue color, sky blue color. There are many red and dark blue colors, right? So when you look into the inner layer, there are gray color and light purple colors are more, right? That means there is asymmetric distribution of the phospholipids. There are many types of phospholipids. Keep this in mind. There are many types of phospholipids. Not only one type of phospholipid is constituted in the membrane, and they are asymmetrically distributed. Very, very important. Asymmetric distribution of phospholipids is a necessary thing for a membrane to function properly. So we'll discuss that later. Okay. So this is all about the fluid nature of the cell, and why it is mentioned as mosaic, just because it appears like an irregular patterns. Just see this. When you look into the cell from upper view, you are having the red as well as a dark blue color and purple color, light purple color. So there are irregular patterns are there. No, they are not symmetrical. They are asymmetrical and irregular patterns are there. So this is how the mosaic tile will be. So just Google it after the class. You'll get to know mosaic tile. So irregular patterns will be there on the tiles, hence they have named it as mosaic model. So fluid in nature and mosaic in nature, hence fluid mosaic model they have named it. So keeping this fluid mosaic model apart, so we are not going to discuss about fluid mosaic model right now in this class. We are going to discuss about fluidity first, right? Next. So to understand about membrane fluidity, we need to understand what are the constituents, chemical constituents of the membrane. It is very important, right? So first one, it's membrane lipids. So the membrane lipids, these membranes contain a wide diversity of lipids, all of which are amphipathic in nature. Very, very important. Amphipathicity or amphipathic meaning contains both hydrophilic hydrophilic and hydrophobic groups. Hydrophilic as well as hydrophobic groups will be there. That means water loving as well as water hating groups are present in a single molecule, and we say it as amphipathic in nature, right? So we are going to discuss how it is amphipathic, why it becomes hydrophilic, why only a head group will be the hydrophilic in nature, why not tail groups. So everything in detail we are going to discuss in this class. Okay, next. So speaking about the membrane lipids, we have three different types of lipids, general classifications. and you have sub classifications too, don't worry about that. The main classification is 3 groups. First one, we know that phosphoglycerides, second it's sphingolipids, and third it's cholesterol. Now phosphoglycerides and cholesterol is very important thing we are going to discuss in this class. We are ignoring sphingolipids as of for this class. If you have out of interest, you can read it, right? Next, so before going to phosphoglycerides, we need to understand about the lipid molecule, the phospholipid, which is the constituent of your membrane. We need to understand about that. Okay, so we'll discuss about this. So we know that generally, we already know this, we have a head group and we have a tail group, right? So we'll just write this structure as like this, no? So this is head group and this is tail group. The same thing we have shown here. The chemistry behind this particular structure, we have given this here. Now, so they have clearly mentioned that this the spherical shape what they have made here, it is called as head group of the phospholipid, and these two are called as tail groups of the phospholipid, right? Now head is called as hydrophilic in nature, tail is called as hydrophobic in nature. See, just remember, I told you from scratch we are going to start. Hence, for most of the students who might know about this, they feel it's already known what he is teaching is it's for CSR net or what. Of course, it is for CSR net only. It is very important. Then only you can understand the factors of affecting the fluidity, right? So you will see the questions also. We are going to discuss certain questions from previous year CSR net examinations, and we are going to analyze why it is important and all, right? Now, so hydrophilic in nature, why this head group is called as hydrophilic in nature? This is my first question, right? So most of the students have to think, whenever they say something, you shouldn't accept it. The very first thing is you have to think by your own. If it is right for you, then only you have to accept it. If it is not right, then cross check with them. They'll give the right answer and they'll make sure that you have understood everything, right? Now, so we know that head group is hydrophilic in nature. They have mentioned it, hydrophilic, water loving. Hydrophilic is called as water loving. Now they have mentioned tail groups. It is hydrophobic, water hating, water hating. Now, so here comes a beautiful chemistry, right? The actual biochemistry of cell membrane. So we need to understand why hydrophilic nature is present in case of head group. Why not it is there in case of tail groups? So tail is nothing but it's a fatty acid, right? So it's a fatty acid. It's a lipid molecule. So saturated fatty acid and unsaturated fatty acids, we are going to discuss it in next slide. Don't worry about that. Now, fatty acid, and above you have glycerol molecule and phosphate group. Now, so based on this, they have asked many questions in CSIR net also, in GATE examination also, right? Now, for example, we know that triglycerol, we have triglycerols, right? So CH2, CH2, CH, right? We have CH, CH2. We have like I'll just change it. I'll change the annotations. Right, I'll write it again here. So anyhow they have given here, so CH2, so and one more CH and one more CH2. So here it is attached with C double bond O, and here also C double bond O, C double bond O, which is attached with fatty acid tails, right? This is called as triglycerol, triglycerol, right? So triglycerol, we know that the normal dietary fat is triglycerol in nature, but remember they have asked many questions based on this. Which of the following constituents is not a component of cell membrane? This was the question. So it has been asked in GATE as well as CSR net also in previous long back ago. Now, so the options were triglycerols. Options for that particular question. The question was, which of the following constituents are not the components of cell membrane? This is the question. So options were like triglycerol, right? And the second option is like diglycerol, and third option is like phosphatidylethanol ethanolamine, PE, and cholesterol. Now remember, Phosphatidylethanolamine I have not yet discussed in this class, but still it is a constituent of membrane. Cholesterol is a constituent of membrane. Out of these two, you need to eliminate one, diglycerol or triglycerol. Now, I told you already triglycerol meaning three fatty acids should be there, right? See here, three fatty acids should be there as a triglycerol. That means the right answer is triglycerol. Triglycerols are never be present in the cell membranes. Remember this. Triglycerols are not be presenting in the, they are not present in the cell membrane. Keep this in mind. Now, what is present in the cell membrane? It is diglycerols. Diglycerols meaning only two carbons are esterified with, esterification will be happening there, esterified with fatty acids. Then one carbon is esterified with phosphate group. See this, one carbon CH2 is attached or esterified with phosphate group. No, so here the very first, let us take it as 1 2 3. So first and second carbons are esterified with the fatty acid, and third carbon is esterified with the phosphate group. Hence we call this particular structure as phosphoglyceride. Phosphoglyceride. Why glyceride? Because glycerol is a backbone there, right? We have CH2 CH2. In this glyceride, two carbons are esterified with fatty acids. That's why we call it as diglycerol. It's a diglycerol, remember. Diglycerols are the major components of the cell membrane. Remember this. Now, so instead of having all this fatty acid, two fatty acid tails and one phosphate group in the glycerol, we say it as phosphoglyceride. It's fine. Now, what makes head group as a hydrophilic? What makes it as water loving? Now, to understand this, you need to understand about what is solubility. So question yourself, what is solubility? Try to answer. Try to get the answer in your mind. That's it. Now, solubility can be explained by different factors. Now, one factor among them is any molecule, always remember this, any molecule which has a capacity to form hydrogen bonding with water. If any molecule which forms hydrogen bond with water, that molecule is said to be water friendly. Always remember this. Then what is hydrogen bond? A question arises, right? So you don't expect this. So based on hydrogen bond, CSR have asked many questions in previous years, right? So they used to ask, like, for example, I'll just write 4 to 5 options to make sure that you may uh, answer it, right? So I'll write carbon, hydrogen, nitrogen. So I'll write in second like carbon, hydrogen, carbon. Third, I'll write it like oxygen, hydrogen, nitrogen. Fourth, I'll write like nitrogen, hydrogen, carbon. Fifth, I'll just write it like oxygen, H, and oxygen. Right. So among five, which of these do you think is a hydrogen bond? So see, in this we have hydrogen bonds also and non-hydrogen bonds also. Which of them do you think which is hydrogen bond? Just look into those and apply the basic chemistry concept and then let me know. Because based on this, the CSR asked questions.